So for our next session, we have the first speaker, Frank Noé from Berlin. His title has changed a little bit, I think, Simulating Signal Transition from Molecules to Cells.
Is it? Yeah. Okay.
Please.
Thank you. Does the mic work?
No.
Okay. Yeah, so I decided to change the talk title and the content a little bit, so let's see how that goes and how it fits on time, but you're watching me closely, so I'm not worried about that. Okay, so first of all, I thought I focus on this talk a little bit on modeling. So trying to think, what are the questions that we want to answer, what are the systems we would like to study, and what are reasonable methods and computational models that would be able to describe what we want to do. So first of all, let's look at the different scales that we're dealing with. So let's look into cells. This is a brain cell in this case, and this is a simulation done by McGuffey and Alcock using Brownian dynamics of a chunk of cytosol, which is actually not from a brain cell. So that's biologically nonsense, this slide, but this is a methodological conference. So let's focus on the scales here. And what we see here is a box of about 100 nanometer edge length. So this is already below the diffraction limit of visible light. So that means whatever is in this box you cannot see with a normal microscope, or you cannot resolve with a normal microscope. You can resolve some of this stuff with super resolution microscopy if you label some of these molecules, and if you either immobilize them or if you do the labeling very sparsely. But basically already here we're in the realm where we can do something with simulation, but it's very difficult to look at this scale experimentally, and often we can only look at things indirectly in experiments. So cytosol is very crowded, and another thing that this movie beautifully shows, of course it's a simulation, but I'd like to believe that it appropriately shows that molecules are sticky. So of course some molecules are really tightly bound and have a specific interaction. Some are kind of transiently sticky, so they like to hang around with each other for short time scales, for let's say sub-millisecond time scales, and then move on. Now let's go into even more detail and look at two of these guys. These are two proteins. In this case, Barnase and Barstar, again, not from the same biological system, so just about scales. And this is an atomistic system now. So here I'm showing the backbone of these two proteins, and this is a water box with about 100,000 atoms. And now we're doing molecular dynamics. Of course, this is something very complex and costly, because in principle we have to compute the interaction forces between all pairs of atoms. Of course, we're not doing that pairwise. There are n log n algorithms to do that smarter than to compute all pairs, but still it's an expensive operation because there are 100,000 particles, so we have to do quite a bit of work in every time step, and every time step moves us only a few femtosecond ahead in time. So it's easy to see that in order to get to something like milliseconds or seconds, it's going to be ridiculously expensive. So because of that, and this is called the sampling problem in the molecular dynamics field, we could basically run simulations of lengths of 100 nanoseconds, maybe a microsecond, something on this order, and that would already be quite expensive. And in this time you can, for example, get an approach of two proteins and they start to interact, but they don't really bind, because that happens on longer time scales. Now, in the last few years there have been some breakthroughs in this field, which I'm going to explain a little bit later. And because of this, and it's basically a combination of computational or hardware advances, but also of mathematical and computational models that help us to solve this problem more efficiently, we can now effectively generate much longer simulations. And this movie is really a reconstructed movie from a kinetic model and I'll explain in a minute what that means. But this is now effectively showing you 0.1 millisecond dynamics, so a factor of 1,000 longer than the previous one, and the clock ticks faster in this movie. And here you basically see that this molecule searches the surface of the other molecule. There's still all the water around that I'm not showing, right? So the simulation still has 100,000 particles. And there are many metastable states, and eventually the molecules find their binding pose, and then they bind, and they stay there for a very long time. Actually, the bound state in this in this complex is like 1 hour of real-time lifetime. Okay, so let's go back up in scales. So of course we don't just have molecules that are kind of diffusing around in the cell, especially with the recent advances in super-resolution microscopy. There are more and more findings that suggest that many of the biochemical processes are very organized, spatial-temporally organized, meaning things are not just kind of well mixed and diffusing around and randomly finding something somewhere, but there is a lot of spatial organization in terms of supramolecular machineries, like this thing here, which is called the active zone. That's actually a huge protein complex, which is partly static in the sense that some of the proteins stay in this complex for a long time. Some of the proteins kind of come and go on milliseconds to seconds time scales. And this is the place where neurotransmission happens in synapses. So these guys here are neuronal vesicles, which are themselves actually complexes of membrane and many proteins. And these are very long structural molecules that probably serve as anchors to these vesicles, and the vesicle can then somehow either travel down this molecule or diffuse to this membrane patch where they fuse. And this system organizes a number of molecules that work together to make this process, endocytosis, happen. So there are calcium channels which open upon depolarization of the membrane, let calcium in, and this triggers a signal which is received by calcium sensors here on the vesicles, which makes them fuse with membrane proteins and eventually undergo fusion. So there is already a machinery in place of many molecules that kind of work together to a certain task. And looking at the last few years of progress in biology, it seems that a lot of biology is organized like that. And we find more and more stuff as we look into more detail into cells. Okay, so all of this organization gives rise to biochemical pathways that are, of course, somehow spatio-temporally implemented in the cell. And this is what we would like to model. So we would like to model in space and time how something like neurotransmission or endocytosis or phototransduction or other signaling processes take place in the cell. And by spatial-temporal, what I mean is I'd like to understand, essentially I'd like to be able to visualize a realistic molecular movie in the sense where is every copy of every participating molecule at any time. And by realistic, what's meant is that the statistics of this movie, of course the movie is nice to present, but it's not the main result. But I mean the statistics of the simulation should be somehow right. When we look at particle numbers, so here are some counts. I don't take the exact numbers too serious. Of course, these have big error bars, but these are counts from experimental studies of proteins in the synapse and in the vesicle, and you see that basically if we want to simulate things on this scale, we have to deal with tens to maybe tens of thousands, but usually thousands of copies of molecules. So this is quite a bit, but if you're used to something like molecular dynamic simulations, that's not shocking. Okay. So, okay. In summary, what I think are the ingredients that we need to model a cellular signal transduction. So, first of all, there's a molecular scale. So, I think molecular dynamic simulations are great to look at molecular scales, meaning something like up to 2 proteins, not too large proteins. These are questions that we can now address with atomistic simulation. And then for the higher levels, so what I'll be calling macro scale in this talk, basically I think we need particles. They need to move around, for example, by diffusion or maybe by some other kind of dynamics if, let's say, non-overdamped effects or hydrodynamic effects are relevant for your problem. They need to be able to interact in some way. So certainly they should not be able to be on the same place in space, but possibly you also need interaction forces, additional interaction forces between them, like long-range electrostatic interactions, for example, something that gives rise to clustering of molecules, etc. And you need some sort of reactions. For example, binding reactions or enzymatic reactions. Okay, so we're in the realm of particle-based reaction diffusion plus interactions, so some sort of forces between particles. Okay, before going there, I first want to go a little bit more into this molecular dynamics world, because this is probably a field that not too many people in this audience are very familiar with, and say a little more of how this speed up in sampling efficiency can be achieved. So first of all, there was a lot of change in our field due to GPU computing. So we have very fast graphics processors now that are very cheap. So for $500 you can basically buy a supercomputer. And these things are getting faster at a very high rate as well. And they're very good codes now. So we can achieve throughputs like 200-300 nanoseconds of all atom simulation per day on one card. But the card is reasonably cheap. So if you buy a cluster with 100 or 200 GPUs, that means you can get a throughput of something like a millisecond per month. And that's amazing compared to what we could do like 5 years ago. That's like a great progress. Problem is, all of these simulations are relatively short. It's not a parallelization, it's just distributing the problem to many computing units. So we need something to put these individually short simulations together to something which describes the long-term kinetics. And there is a whole range of models, kinetic models, that do this job in some way, and this is also what my group is doing for a good part of their work. And one of them that is very well known is Markov state models. So Markov state models is an approach in which you partition the state space, the molecular state space, into substates. You then start simulations from different substates, short simulations, and you record the statistics of transitioning to different substates and estimate a Markov chain from that. And with this Markov chain, you basically predict the long-term behavior. That means the stationary properties, equilibrium distribution, but also the rates between long-lived states. And if you do that well, you can basically, if you're doing aggregate simulations worth 1 millisecond, really describe millisecond kinetics or even slower kinetics. There's a lot of theory that I'm not going to go into in detail. we have a program, PyEmma, which basically does the Markov modeling. That's an analysis package that you can give basically loads of MD data and build the models in this way. Yeah, there's this children book, Jim Knopf und der Lokomotivführer in German. And there the locomotive is called Emma. This is why we decided to have a train. I'm sorry? Yeah, Python Emma, exactly.
How large the space of Markov space should be to be realistic?
Yeah, it's surprisingly small. So the trick behind this is basically all of these methods can be termed as Galerkin projection methods of the Markov operator. Now, in systems like proteins, you typically have some slow timescales and many fast timescales. And so what you need to do is to get a good approximation of the dominant eigenfunctions of the operator. If your system is metastable, these eigenfunctions are piecewise constant and then have steps. And so that means ideally you can describe one relaxation process with two states. But a typical number is a few hundred or a few thousand states for a protein. So it's not astronomical. It's not something like, don't think of this as like PDE discretization or something like that. It's completely different. Also, we have one parameter that we can turn here, which is the lag time. The lag time is the time step made by the Markov chain. So with the Markov chain, we're trying to approximate the statistics of going from state I at time t to state j at time t plus tau. And this tau basically is a parameter with which I can basically kill many of the fast processes. And this helps me a lot with dealing with relatively coarse discretizations. Okay, so that's the approach here. Of course, now what I haven't really said is how do we even find states? Because if we just start 1,000 parallel simulations in the same state and it takes a minute to get here, then I'm never going to get there just by hoping for the best. So I need to do something about that. And one approach here is so-called adaptive sampling. And the idea is basically you start parallel simulations, you simulate, and then you look at the history and you select structures for the next round of simulation. In this selection step, you could favor structures that explore new states, for example. So you could favor pushing the simulations out, but by pushing I really mean you just select starting points in a smart way. You don't bias the individual simulations. Those simulations here are still unbiased equilibrium dynamics, MD trajectories. The only thing that's not in equilibrium is the starting point. Okay? And with this approach, if we go back to Barney's pasta, there's two protein complex that I've shown you, which I told you has a residence time of like 1 hour in the bound state. We can, on one hand, we can find the bound state from dissociated states very quickly. So these are microseconds here. So we go from a high distance from the bound state to the bound state in many runs. But we can also, when we start from the bound state, dissociate, which means that this doesn't happen in one step, but in many little steps that we can somehow use as milestones. Okay. Overall, we get, in this example, several thousand association and dissociation events. And by associated, I mean conformations where the two molecules are somehow touching each other. And we get about 90 binding events. That means from some conformation where they're touching each other somewhere to the really bound pose, and about 10 unbinding events. So here's the statistical bottleneck. Okay, so last step, we need to reweight the trajectories somehow. I told you the trajectories are individually unbiased, but their starting points are not taken from equilibrium. So that means I cannot simply do something like histogramming or counting or anything that assumes that I'm sampling from equilibrium. I cannot do at this point. This is why we use a Markov chain, but we want to additionally enforce that this Markov chain fulfills detailed balance because everything is in equilibrium here. So I'm solving this maximum likelihood problem. And this can basically be solved in an iterative way, where I take a starting guess for the equilibrium probability. I update it with this update equation, where the W is defined here, and that basically contains the transition counts. So the statistics are how often I went from I to J and J to I. Once I'm done with this iteration, so once this has converged, we compute the transition probabilities in one step with this equation. Okay. So now we do this for the trypsin benzamidine, Barney's bar star problem. And what we've done here is to run an aggregate of 2 milliseconds of molecular dynamic simulations, some of them adaptive, some of them non-adaptive. These are about 4,000 trajectories, and they were generated on many, many GPUs over some time. And here is a Markov model which summarizes the results. This is actually using a hidden Markov model that I haven't explained, but it's the same idea. Okay, so here we have basically an unbound state. So these are states where the two proteins are either not bound or they interact just very weakly. Then we have some states here that are non-native, I would say. So the native state is defined here by this green guy binding into this cleft. These states are states where the other proteins is on the wrong side. The bar star protein is on the wrong side. Then we have states where the protein is basically bound in the binding site, but is the wrong way. We have something like a transition state or a pre-bound state, if you like, where the binding is almost correct, but the one protein still needs to turn in order to bind, really. And then on the bound state actually has two substates between which it fluctuates. And this happens on a microsecond time scale. The whole binding process takes about 100 microseconds. This process is on time scale of several microseconds. The unbinding takes, as I said, the whole dissociation takes hours. The partial dissociation to these substates here takes about minutes. So on a time scale of minutes, you fluctuate in this bound state, then you sort of unbind to this denatured state that are still where these proteins are still touching each other, but then rebind. And this happens many times before you really unbind. So this is the kinetics of this protein, and this is now a kinetic model. Okay, and this kinetic model is now something that we can use on a higher scale, on a particle-based reaction-diffusion simulation scale. Now, of course, this is a little bit too complicated and maybe much more detailed than what we need at this level, so we can coarse grain this. We can decide to aggregate states here that exchange quickly enough so that at a long time scale we don't care. So, and then depends what time scale we're talking about, but we can coarse grain this either to a 6-state model or to a 3-state model where we basically just distinguish dissociated state and counter complex, which is all of this here, and the bound state, which aggregates these two states. Okay, so this is a way of getting sort of microscopic kinetic models from microscopic simulations. that we can introduce into macroscopic simulations. One note here, most of the rates estimated from this model are quite accurate. By that I mean they compare well with experiments that are trustworthy and also statistically precise. But what is not precise at all is the dissociation rate, because there are some slow steps in here that we haven't sampled very well, even with these techniques. We're basically pushing it to reach these time scales, and the dissociation rate has an uncertainty of 6 orders of magnitude. Yeah? So it's like something between a minute and a week or so, which is a little bit frustrating, of course. So even these simulations are, in many cases, not sufficient to get very reliable or sufficiently precise models. But there are ways to improve that. So especially if you have slow steps, you can combine direct molecular dynamics simulations with generalized or biased ensemble simulations. So these are basically simulations that somehow change the potential or change a thermodynamic parameter like the temperature in order to overcome high energy barriers better. And we have developed a way that uses the Markov chain maximum likelihood estimation and modifies it in a way that you can include data from all of these biased or generalized ensemble simulations. And so you basically estimate a Markov model on multiple ensembles, and they're coupled with one another by the fact that you can reweight points between different ensembles, because you know the stationary densities point-wise. And with that trick, you can actually get dissociation rates with relatively high accuracy and also with lesser effort. I'm going to skip this, but this plot here shows that you can estimate the dissociation rate of a ligand from a protein, which is just fast enough that you can also do it directly with only a small percentage of the data with the same precision as if you would use the full data. Okay.
Sorry. How do they compare to experimentally measured association dissociation rates?
In this, so I mean the example that has an uncertainty of 6 orders of magnitude, obviously the experimental value lies in the uncertainty window, which is would be pathetic if it wouldn't. In these cases, usually I would say we are within an order of magnitude in the cases that have been sampled well. There is still a systematic modeling error here. We're using an empirical force field, so that could be arbitrarily wrong. And there are some effects that we know cannot be described by these force fields, such as polarization. So it could be very wrong. But in most cases that I've seen, like protein-ligand dissociation, protein-protein dissociation, binding, unbinding, folding, unfolding, I would say an order of magnitude in the rates is about what we can expect if the system is well sampled. Often the sampling is the bottleneck. Okay, now let's go to interacting particle reaction diffusion. So this is what we want to do in the macro scale. So we have particles A and B. They can diffuse around, form an encounter complex, and undergo a reaction to particle C, as one example. Or you could have A goes to B, or A plus B goes to C plus D, all of these versions. But we're basically going to limit ourselves to binary reactions at most. So anything that's more complex than binary has to be broken down into binary steps. Particles can diffuse around. So basically, by default, we assume that there's Brownian dynamics, although there could be other dynamics, such as non-overdamped Langevin dynamics, but for now, let's assume Brownian dynamics for these particles. It's Brownian dynamics in an interaction potential because we want to allow these two particles to interact with one another, even if they're not in reaction distance. And we have reactions. So first of all, since we have potential, at this point, we can write this reduced potential, and then depending on the thermodynamic ensemble that we want to simulate in, we expect a certain density of states. So that's nice because that gives us some thermodynamic reference that allows us to check if the implementation of the simulation is correct. About kinetics, kinetics is a little more difficult. So if we have non-interacting particles, meaning particles that don't have an interaction potential but just react with one another, then, as already described this morning by Alexander, we can use this equation that's appeared in this Erban and Chapman paper here to relate the microscopic and the macroscopic rates. And one thing one has to keep in mind here is that usually what we get from an experiment is a macroscopic rate, so something like this. Yeah, so this is an association rate, which is in mole and second. So it's a concentration-dependent association rate. And it can be realized microscopically in this scheme. And if we choose to do that, then we have a number of parameters that give rise to this macroscopic rate. But this is not a unique choice. What is pretty unique is the diffusion constant, because you can measure that independently of this process. So let's say we have the diffusion constants as well as an input. But then you still have this reaction radius and this microscopic reaction rate within the reaction radius. And these two are to some extent can be chosen. So we can make the reaction radius bigger and the reaction rate smaller and still get the same macroscopic rate, as long as you're not looking at anything else than this relation at least. So this is something to keep in mind. So usually what we do is we choose reaction radius to be, a reasonable molecular interaction radius in solvent, which is like 1 nanometer distance, surface distance. So 1 nanometer plus particle radii, and then determine the reaction rate. But all of this is no longer valid if we have interaction potentials. Yeah, if we have interaction potentials, then this equation doesn't hold anymore, because the interaction potentials create something like excluded volume, maybe. We do not have a general equation for this case. So what we can do is to do short benchmark simulations in order to parameterize for the microscopic rate in order to implement a certain macroscopic rate. And if there are simple interaction potentials, like radial interaction potentials, then I guess you can still do that numerically somehow. Okay, implementation, basic implementation is simple. We do Brownian dynamics with isotropic diffusion. We have a reaction time distribution in each time step. So we have to choose time step small enough to the fast compared to the fastest reaction, the largest reaction rate. And then we discretize this stuff. Now, all of this has certain problems. So, for example, this discretization scheme does not satisfy detailed balance. And in general, we have to be quite careful with detailed balance when we execute reactions. A plus B becomes C, and let's say this reaction is reversible, then we have to take care where we put A and B on the return step. So these are things that have been realized in other reaction kinetic schemes long ago, of course. And here we just have to sort of adapt these methods or develop new methods because our particles are interacting, so the schemes are slightly more complicated than the cases for which this has been solved already. Okay, so this approach is implemented in the READY program, which was originally written by my grad student Johannes Schöneberg, who is now in Berkeley, and he's become a single molecule experimentalist now, at least for his postdoc. Let's see if he returns to theory. But he was a great guy, and he basically implemented this whole thing by himself. This movie sort of shows what it already does. So diffusion. This is kind of predator-prey simulation where the blue guys are the predator and the green stuff is the food or the prey. And these red particles here have some interaction potentials that makes them sticky, so they like to stay together. And so it takes a while for the blue guys to enter the D here and eat all of the food that has accumulated here. Ready 1 was written in Java, and Java is not a high-performance computing language, but it's a good prototyping language. This is why we have chosen it for the first version. We are now rewriting Ready in C++, with some C obviously, and then also an automatic way of generating Python API from the C++ headers so that you can interface it from a Python notebook or a Python script. Okay, so what can you do with Ready? Obviously simulations like this. This is just basically a chunk of cytosol where you can simulate the crowding of particles. But because these interaction potentials can be quite general, you can also build larger molecules. So you can build chain molecules where you attach multiple particles with springs to something like a filament. So you can model filamentous molecules. You can have proteins diffuse on surfaces or spheres or other geometric objects. And yeah. Right, so what's the status of this project? So currently we're writing Ready 2. Ready 1 is pretty much tested and usable, and there are also some tutorials online, so you can download and try this. It's no longer supported in the sense that we're not working on the code of Ready 1 anymore, because now we want to focus on the new implementation, which is in C++ and has different kernels, and we're currently working on the GPU kernel, so we want to get that running first, because that allows you to even do some simulations just on a desktop with a nice GPU. Rough benchmarks, so that you have an idea. If we choose a 0.1 nanosecond time step, which is a reasonable choice for small membrane proteins, so that would be a shorter time step if you have proteins in the cytosol which diffuse faster. Then you get something on the order of a couple of milliseconds per day per GPU. So this is not something with which you can do metabolism over many hours, at least not very soon, I would say. There are a number of ways how we can push this, I think, but this is about what we get at the moment. So you can do a simulated real time.
Of roughly something like, you're talking about Ready, right?
Oh, 20,000 particles.
Oh, sorry.
Yeah. So you can probably get to a few tens of milliseconds if you have less particles. So that means fast signal transduction processes that occur in the timescale of hundreds of milliseconds, 1 second maybe, neurotransmission, phototransduction, all of that stuff. That's within reach. Cell division is not, at least not in the moment. Okay. What are open problems that we're working on? So one thing we'd like to do is integrate event-based integrators to some extent. Basically, this is I'd like to think of this as a molecular dynamics code with reactions. Yeah, so basically, it's kind of a thing that lives on discrete time steps. But there are some processes for which this is horribly inefficient, and we really want to make use of these event-based ideas like first passage kinetic Monte Carlo methods. And one example is when we have slowly diffusing particles in a membrane and much quicker, much faster diffusing particles in the cytosol. Now, if you would just use a fixed time step for everything, the same fixed time step for everything, then these guys determine the time step because they're fast. But that means that we sort of slow down everything by a factor of 100, and that's really not necessary because these things are also dilute. So we should be able to take longer time steps if we knew, for which particle we're doing what. So basically what we can do is to draw protective domains around these particles and decide how long is it going to take until this particle will exit this domain, and then sample its exit point, which is sort of the simplest version of first passage kinetic Monte Carlo or EGFRD. And then basically we can schedule these particles to jump long steps ahead and prevent them from slowing down our simulation. So this is something I'd like to implement. Another thing that we're working on is reversible integrators that respect the macroscopical statistics. That means we want to get a reaction scheme, A plus B becomes C, and reverse. That is, on one hand, fulfilling detailed balance with respect to the interaction potential, and on the other hand, basically gives you what you put in. So you want to get certain binding affinities or binding free energies and macroscopic on-off rates out of the simulation. And in the dilute limit, so in the limit where not everything is crowded and everybody is interacting with everybody, but let's say where you have just two particles in your simulation, you want to get that statistics. So this is something that the integrator should be able to give you. And then if you have crowding, you may have changes to those statistics, because basically that will affect the chemical potential. Another issue that's very important for us is membranes. We want to do endocytosis, and it's clear at this point that I will actually not come to the applications part of the talk, but that's no problem. So how do we do something like membranes? What I've shown you before, there were membranes kind of as surfaces, fixed surfaces, so spheres or planes, and they were not changing in time. But for endocytosis, so endocytosis is the process where the cell uptakes something from the outside by forming a vesicle out of the membrane and creating fission and moving this vesicle inside. In order to do that in Ready, we want to approximate the membrane by particles. So basically a sheet of particles. And now each particle is not something like a lipid molecule, but it's actually a chunk of the membrane. So it's many lipid molecules. And this model here is a dumbbell model. So it basically has two kind of lipid proxy molecules that sort of mark the membrane normal, and the neighboring dumbbells are interacting with each other with Morse potentials. Basically what my postdoc Mohsen has done is to parameterize this model with the Kahnham-Helfrich free energy density. That's basically the correct expression for membrane bending energy in the small bending regime. And you can simulate this model and get the equilibrium fluctuations, and there's also a way to check that you're simulating the right fluctuations. And because we have Morse potentials and membrane fluidity, we can also simulate things like endocytosis. So this is actually the uptake of a nanoparticle by the membrane just by hydrophobic forces. So there's nothing pulling this particle. just a hydrophobic particle that we put on top of the membrane, and it likes to wrap itself in the membrane because that's energetically favorable. And you'll see here, you see the membrane fluidity quite nicely, and eventually the neck will become thinner, and at some point it will also, not in this movie, but at some point it will undergo fission. Okay, so I'm gonna jump over the application part. And come to the credits. So yeah, there are many people in this picture that I should thank. So basically, many of them are involved in molecular dynamics simulation and Markov state modeling, and yeah, here and here, Mohsen Sadeghi, Moritz Hoffmann, Luigi Spailo, and Chris Fröner. This is the Ready team. So these guys are now in charge of Ready. And our aim with this program is basically to make it as open and flexible as possible. So there are limitations to the size of a code that a group can sustain over a long time. We're very aware of that. So what we want to do is to create a platform that makes it easy for everybody to not only use but also extend it to insert the integrators that you want, and if somebody wants to do hydrodynamics, to insert that, and we make sure that there is a framework that you can do that efficiently and you don't slow everything down by inserting some code. But we don't want this to become a massive monolithic code. It's more of a framework. Okay, thanks for your attention.
We're somewhat behind. Do we have one question, perhaps? Yes, over here.
If you have excluded volume because of the interactions, would you set the reactive radius to something larger than the, like, twice larger, or what?
Yeah,
what,
you can.
or is it just the parameter you tune? How is that?
Yeah, so so we have an expression for that in the in this reversible integrator. So when you ask for the scheme A plus B becomes C to be reversible, it turns out you need to add a certain parameter that enlarges your reaction volume, and this parameter basically depends on the integral over the Boltzmann density created by your interaction potential.
If delta T goes to 0, that would go to 0, or does that remain there?
Then it goes back to the radius that you need to sort of this Urban-Chapman solution. Yeah.
Okay, we need to move on. Thank you very much. Thank you, speaker again.